Can't hear you, ma'am. Ma'am, you're muted. Can't hear you.
Okay. Yeah. So, okay, that was my fault. I had not unmuted myself. I hope you can hear me now. Um, yeah. Okay. So I'll share my screen and we can begin with the session. Somebody's trying to join in. So hello everybody, welcome to probably the final session, final discussion session for your course Neurobiology. Although there are chances that we might keep one extra session for the final discussion and summarizing of this course, but we have not decided on a session or a date as yet. So this might be your last session, at least the previous year questions that we have been discussing. We have it till week 4, so that will be the final one today. I am Arveen. For those of who are new to this session, I am currently a PhD scholar at Department of Biological Sciences and Bioengineering at IIT Kanpur, where Professor Gupta is also a faculty at our department, and this is one of the courses that I have taken from him during my coursework itself. Currently, I'm a TA for this course. Right. So what we are going to do right now is that we'll divide our session into two parts, like we have been doing for this course. Right. The first part, we will be discussing some previous year questions that are pertaining to this week's lectures, that is week 4. Right. And in the second part, what we'll do is a recap of whatever has been taught to you during this week by Professor Gupta. Right. With that, let us just begin the course. Yeah. Okay, I will read out the question. I will read out the options. I will take a slight pause for those of you who actually want to attempt the question, and then we'll come to the answer and discuss upon why that particular option could be the answer, right? So the first question for today is a single action potential usually releases neurotransmitters from one synaptic vesicle. Is the statement true or false? Right. You were introduced with the concept of synapses in the week 3 lectures that were given to you, right? Shortly after that, there was talk about individual kinds of synapses, whether it is electrical or chemical. Over here, chemical synapses are being hinted to, right? Because that is where neurotransmitters are involved. So the answer for this particular question is going to be false, right? Why is it going to be false? Because usually, there are multiple action potentials that are coming continuously through the first neuron. And even if we do not account for that, even a single action potential will basically not go and attack one particular synaptic vesicle. It is going to, take that action potential and that conducting action potential at the end of the presynaptic terminal. There will be multiple synaptic vesicles or these vesicles that contain neurotransmitters in them, and multiple of those vesicles will actually go and fuse at the end of or the terminal of this particular neuron, right? It will not just be one synaptic vesicle that will go forward, right? So the action potential can cause release of neurotransmitters from thousands of synaptic vesicles, right? That is what you need to remember. There is like a single one will not go and fuse, and even if it does, it will not be enough to generate, an EPSP or post-synaptic potential, whether it is excitatory or inhibitory in the post-synaptic neuron, right? So multiple such vesicles are required for actually generating a response in the in the neuron that is following the one that is coming after this presynaptic neuron, right? So yeah, yeah. The second question is also from synapses, right, and from chemical synapses. This question says, in chemical synapses, communication happens by the movement of charged neurotransmitters from one neuron into the cytoplasm of the second neuron. Is this particular statement true or false? Right? So it is a longer statement. There are multiple things that are being said over here. You need to stop and think about all the information that is given in this particular sentence, and based on that, you need to say whether it is true or false, right? Yeah. So when we talk about this particular statement, let's just break it into what all information it is giving us. Okay. It is saying that we are talking about chemical synapses, right? And when in chemical synapses, we talk about communication, it is by movement of charged neurotransmitters, right? Now, this part is also true. From one neuron to the other neuron. That part is also true. Now it is saying what is the medium of movement from this one neuron to other neuron is that one neuron will basically release its neurotransmitters into the cytoplasm of the second neuron, right? Now this is a part that is actually incorrect. So that thus our statement is false, right? Why is it false? Because what will happen over here? The first neuron or the presynaptic neuron will actually release neurotransmitters, but they are not going to go directly into the second neuron cytoplasm. They are going to go in a space that is present between these two neurons, which is known as a synapse or synaptic cleft, right? And from that synaptic cleft, these neurotransmitters might be taken up through the receptors that are present on the surface of the post synaptic neuron, right? So this is what will happen. The communication happens via release of neurotransmitters into the synaptic cleft, right over here, not into the cytoplasm directly, where they bind to the receptors on the membrane of the second neuron, these receptors, and they do not directly enter the second neuron, right? And not even indirectly, right? Because they are going to go and stimulate these receptors, and the downstream signaling machinery of these receptors are actually going to take care of transmitting that action potential forward, right? It is not really the case that these neurotransmitters are going to interact with these receptors and then that is going to take them inside this particular neuron. No, right? So that is just, they are just building an interaction at this particular terminal, and that interaction is stimulating this particular neuron, right? The postsynaptic neuron. So question number 3 is, proprioception is a sense of which of these? Posture and movement of the body parts, temperature, itch or thirst, right? So which one of these correctly describes proprioception? Okay. So proprioception is basically a sense of posture and movement of body parts, right? So the fact that when you are sitting, you are aware of where your body parts are or where your hand is pointing, right? Or if I'm speaking to you, I'm making some hand gestures. It might be subconscious, but if I try to focus on it, I will know, right? That where are my hands? What are the kinds of movements my fingers are doing, right? I don't really have to look that okay, I am holding up two fingers, right? I can be holding up two fingers in like far away from where I can actually see my hand, and still I know that I'm holding up two fingers, right? So that kind of perception that is present from within our body parts or organs, and it is giving a some kind of a feedback to our brain. That is why our brain knows that this is happening, even though it is not constantly monitoring it, right? That is known as proprioception. So it is the ability to sense the posture and movements, right? Now, question number 4 is, which of the following is most commonly thought to be involved in the signaling of a reward, right? Is it dopamine or serotonin or acetylcholine or glutamate? So which of these is most commonly associated with signaling a reward behavior? So now, if you remember, these were the names introduced to you when Sir was speaking about neurotransmitters, right? So these are all neurotransmitters, and dopamine and serotonin, when those were being introduced, uh, along with the noradrenaline in Sir's lectures, he also mentioned them to be neuromodulators, right? Because they are not just involved in these junction to junction activity, right? They are also involved in higher level activity in the sense that uh, the kinds of cognition or the sense of reward or pleasure or sadness that is going on in your brain is actually affected by these particular neurotransmitters or neuromodulators that are working at a very high level, right? Not at one particular junction, but at multiple regions in the brain, right? So oh, at that time, we did talk about dopamine, right? Now, dopamine and serotonin, if you move further, like away from this discussion, and like, I don't know the kind of literature you guys have read or the kind of social media also that you follow. But a lot of even non-neurobiology people are aware of both dopamine and serotonin because these are known as happy hormones, right? So these are basically the ones that are responsible for the sense of happiness in our body. And of these, dopamine specifically is the more potent happy hormone, and it is also responsible for the signaling of reward, right? So whenever, so for example, you had worked really hard for a particular exam, and then you are seeing that you are getting an outstanding grade in that exam, right? You will be ecstatic because of the kinds of endorphins that are going off in your brain, and dopamine will play a huge part in that, right? So the answer for this one is going to be dopamine. It is a major neurotransmitter or neuromodulator that is responsible or involved in the signaling of the reward, right? So, yeah, so this is the structure of dopamine. Although you don't have to worry about the structures, I think, as of now, for the exam point of view, if any one of you have registered. But for your knowledge, this is the structure of dopamine, right? Then we come to question number 5, which says rods respond equally to all wavelengths of the light. Is the statement true or false? Right? So, a hint is that you need to remember that particular map that had a lot of colors, and there were three kinds of rods, and three kinds of cones, and there was this other light receptor or photoreceptor which was known as rods, right? Those four kinds, all of them. Okay, I will say too much if I describe it so much, right? So there was this graph, right, based on which you can answer this question. So the answer for this one is actually false, right? Why is it false, and why I stopped speaking earlier was because in that particular one, like there was this graph. I think I've kept it. Yeah, this one, right? So in this, you remember these three kinds of cones: the blue cone, green cone, and red cone, depending on where their excitation peak is lying, right? So in the green cone, no, the excitation peak is almost in the green region, right? In the red cone, it is towards this yellow red region. In the blue cone, it is towards this purple blue region, right? In this particular schematic only, you can also see that rods have an excitation peak at around 498 nanometer, which means that it is not responding equally to all wavelengths of light, right? That it will respond more to this blue colored wavelength, blue green wavelength than other wavelengths. But since the peak is like the curve is extending for a longer time, right? For example, in the blue cone, there is no kind of excitation at all when it goes into this far green or the yellow red region. Right. Similarly, for the red one, there is no excitation at all in this blue region. Right. So, uh, although these other ones are specific and they do not overlap into other regions, rod have a more wider kind of they are spread across most colors, but that doesn't mean they are responding equally to all the colors. They are responding maximally to this blue color, right, where they have their peak at around 498. So rods also have a tuning curve which peaks around 500 nanometer wavelength, or to be more precise, around 498 nanometer wavelength, right? Then we come to question number 6, which says olfactory stimuli may have less spatial information than vision. Is this true or false? So it is a simple question. This is, of course, true, right? Because in vision, there are the kind of stimuli you get, you can constantly locate where, because you are constantly seeing, you can locate the spatial or organization or orientation of various objects that you are actually visualizing. In case of olfaction, because at one time you will be able to actually perceive only one kind of odor properly, right? If it were mixed with another odor, you will probably not be able to distinguish, or right, if it was immediately presented to you, both the odors at one time. So they do not really have a very good spatial information, right? So if, for example, I was just inhaling one kind of odor, and only one kind of odor was stimulating my senses, and I could maybe figure out, okay, I think it is coming from my right. But if there were multiple odors that were present, I will probably not be able to figure out which odor is coming from where. Right. So in that sense, the spatial information that it provides is much less than the visual stimuli, right? Because visually, if I'm seeing that there are multiple colors, I can still look and I can see, okay, green color is being emitted by that plant over there, brown color is being emitted by that door over there. So I can still visualize, right? So, yes, we have relatively less information about the location of olfactory objects compared to the visual objects, right? Now, coming to question number 7, suppose we are doing electrophysiology experiments to measure the responses of projection neurons to odors. We expect to see the smallest change in the observed temporal pattern of spikes for which of these? Different trials, different odors, different neurons. So this is a good question. Because I think it was not directly discussed in the class, but the concept behind this was discussed, right? Okay, maybe you can go through that statement again and try to figure out the answer. Otherwise, I'm going to obviously explain it to you. Okay. So what are they saying over here? They are doing an electrophysiological experiment to measure responses of projection neurons, which means that there are basically electrodes that are present inside the projection neurons, and they are basically measuring when these neurons are being stimulated, right? When they are giving an electrical response, right? So if we have those projection neurons pinned up with these electrodes and we can see their responses in which of these patterns will we see the least change or the smallest change in the temporal pattern of spikes, that is that how they are changing with respect to the time at which the stimulation was done, right? So is it different trials or different odors or different neurons, right? So if you remember, this basically depends on odors and neurons, right? Projection neurons, which particular projection neuron is firing, how much a particular projection neuron is firing is dependent all upon which neuron we are looking at and which is the odor that we are giving, right? So these two factors are controlling that. If we are altering these two factors, these are anyway variables, right? So if we are altering them, then there will be many changes that we will observe in those projection neurons, right? But if we are just doing different trials, what does it mean? That, for example, one particular odor was giving at one particular time and the response was taken. Now, again, it was given after some time and again, response was taken. Like similarly, the similar experiment is being repeated, right? Continuously. That is what different trials means. So the least or the smallest change that will be observed in temporal pattern of spikes will be for this particular one, that is different trials, right? So why? Because temporal pattern will depend on the odor and the neuron that is being measured, right? Different trials or repeated experiments with the same odor and neuron should cause the least change, right? For example, when which was that when the sensory response was being studied by you guys, or it was being taught by sir, right? In that you understood the concept of adaptation, right? If constantly a particular stimulus is being given again and again, either it will remain the response will remain same, or after the continuous stimulation, after a while, what will happen? The response will diminish, right? Why will that happen? Because now your body is adapted to that particular stimulus, right? So that is the kind of reasoning you can do before answering such questions. Okay, which of the following is the least accurate about visual perception? Is visual perception is it that visual perception involves many regions in the brain, or visual system ensures that the visual perception is identical to the image formed on the retina, or visual perception can be affected by the prior experience, or artificial stimulation of photoreceptors can trigger visual perception? Now, again, this is one other slightly tricky question. So I would want you to go through all the options again, and I will pause a while. Yeah. So let's discuss the answer for this one. The answer is option B. That is, the visual system will ensure that the perception is identical to the image formed on the retina. Right? This is not actually true. Right? When is it not true? In examples such as this one, right? So we'll come to that. So the visual perception involves many levels of processing, right? We are not talking about visually actually seeing. It is visually about what we are perceiving, right? So sometimes we just don't see a black and white object as a black and white object, right? Even when we look at clouds, we sometimes try to make, shapes, like observe what shapes these clouds are in. So they are not actually present in a shape, but that is how our brain is trying to process it, right? So the visual perception involves many levels of processing and abstraction by the brain, and therefore the perception can be different from the actual image, right? As is exemplified by various illusions. One of the illusion examples I've kept over here, right? So in this, for instance, there were three of these, partial spheres that were present, and then three of these V's that were present, right? But when we actually look at this image, it looks like there are two triangles that are lying on top of one another, and that is why such a representation is coming out, right? So actually what is given or what is drawn is not exactly that, right? But that is how our brain is perceiving. So thus this one is the correct option, right? That the perception is not exactly identical to what is the image being formed on the retina, right? Then we come to question number 9, that is, pheromones are most likely to be involved in which of the following cases? The attraction of female flies to male flies, or attraction of female flies to humans, or attraction of female flies to flowers, or attraction of female flies to bananas. Okay. So the answer to this one is attraction of female flies to male flies, right? Now, what are pheromones? These are special chemicals that are basically used for, the coordination within the same species, right? And in insects especially, we do know that definitely there is an existence of these pheromones, right? And pheromones are involved in communication between members of the same species. Now flies are the members of the same species, whether they are male or female, but flies and humans, flies in flowers or flies in bananas are all different species, right? So definitely in this case, just by the definition of pheromones, we can rule these three out, right? And the only remaining option will be our answer. And over here, like, it is not important that pheromones will only communicate between different genders of the same species also, right? Even the same gender, communication can occur via the aid of pheromones. For example, in honeybees, right? When people are, they communicate like where the nectar is, or what is the, which are the combs that have to be fed, which is the comb that has to be made the queen bee and fed according to that, right? All this is done via the communication through hormones, through pheromones, sorry, not hormones, right? Yeah. Also, the insects have a different kind of olfactory, junction that is reserved for, olfaction of these pheromones, which is known as the vasomotor area, right? It is present next to the place where the other odors are actually smelt. Right, we'll study about that when we come in the discussion session. Right. Now, question number 10 is, glomeruli are present in which of the following? The olfactory bulb, the olfactory epithelium, antenna, or mushroom body? So, obviously, we are not talking about the glomeruli that are present in the kidney, right? So, we are talking about the brain over here. And we are talking about the olfactory system of the brain, right? In this particular olfactory system, glomeruli are actually present in the olfactory bulb, right, in the vertebrates. So, olfactory bulb includes various glomeruli which receive sensory information from the sensory neurons, right? And for example, like there is this olfactory epithelium in us, right, where all these cilia and mucus and all are present that will actually go and bind to the odorant molecules, and it will transfer, signal into this olfactory bulb that is present over here. So how will it transfer? Because these sensory neurons are actually going up to regions in this olfactory bulb, and these circular regions are known as glomeruli, as is mentioned over here, right? Now, in the glomeruli, these will come in contact with different, neurons that are carrying this information to the cortex inside the brain. These will be the tufted cells or the mitral cells, right? And these junctions will also occur in these glomeruli that are present in the olfactory bulb. Okay. So those were the 10 questions from previous year's assessment for this particular week, right? So that is it for this part at least. We'll jump on to the discussion session, right? So previously, like up till the last week, how far along I had discussed is the generation of an action potential, right? That part I had discussed, but we were going to talk about the conduction of action potentials. That is a very short part that was remaining pertaining to that action potential unit, right? That I'll finish maybe in the first 10 minutes right now. And thereafter, what we can do is we can continue with the rest of the discussion, right, which is pertaining to the synapses, both the introduction of the synapses, what individually the electrical and chemical synapses are, how they are functioning, how, they are different from one another. And finally, we'll move on to the sensory part, how the sensory system works in our body, and that is like going to be how we are going to end the session, right, with the final discussion about how our sensory perceptions are working with respect to all the major senses that are present in our body, right? So with that, let's just begin with this particular part as well. As I said, there was a brief part that was remaining when we were discussing about this action potential and how it is being transmitted forward, right? So, what the part that we had actually skipped or missed at the end was this part of where the action potential is actually being generated. So let me just take that pointer. Yeah. Of where the action potential is generated, right? And we saw that the action potential was actually being generated at the axon hillock. Now, what is the axon hillock? If you see, this is a structure of a neuron, right? This is the cell body, this is the axon, and this is the near the cell body are the dendrites, and over here is the terminal region of the axon, which again is sprouted into multiple branches, right? So the region where the cell body is actually connected to the axon, or this long basically length stem that is actually connecting one cell body to the next cell body, right? This portion is known as axon hillock, right? And this is actually the portion where action potential is actually generated, right? Why is this the portion where it is generated is what we are going to see now, right? So what happens is that okay, I think I don't have that part over here. First, let's just discuss that then, right? So, why is an action potential generated at the axon hillock? Because over here, the threshold for the generation of action potential is relatively lesser, and the number of sodium and potassium ions, ion channels are more, right? So there could be more transfer of ions into and out of cell, and the threshold can be achieved because the threshold for, attaining an action potential over here is lower, right? So that is why this is the most common place where basically this action potential will originate, right? And that is what happens also. So if we zoom into this region near the axon hillock, and in the axon hillock and near it, we will see that generally we know that the resting membrane potential of a neuron is usually negative, right? In humans, it might be around minus 70. In squid axon, it might be closer to minus 65 or so on, right? So over here, we can see inside of this membrane is negative. Outside, the extracellular fluid is positive in comparison to this, right? And the charge will be lined along the two, ends of this particular membrane, right? Next, what we can see is, uh, yeah, so this is what I was saying, that because the hillock has the high density of voltage gated sodium channels, right, that is where the action potential will actually be generated. Also, what will happen is that in this region, right, there is high density of sodium channels, so more of sodium can actually come in, right? Why will they try to come in? Because the concentration gradient, according to the concentration gradient, they are lesser inside. Also, according to the electrical gradient, there is more positive charge that is present inside. More negative charge that is present inside. So that will also pull sodium ions inside, right? So both the concentration and electrical gradient will favor movement of sodium ions from this channel into the neuron, right? So that is when there will be depolarization that will start happening, and as soon as this depolarization reaches a particular point, a threshold, after that action potential will be generated over here, right? So this is the region of depolarization. Now, what will happen after this region is depolarized? Action potential will be generated over here, but it also needs to move forward. Right now, how will it move forward and where will it move forward, right? There are two sides it can go to. So it needs to decide which direction it will take, right? For that, what will happen is that like for example, this first state that we were talking about was the resting state, and this second was the initiation of the action potential. Now, when it propagates, it will propagate along the axon, right? Why will that happen is because this, if it goes towards this right side, right, over here again the density of the channels is more than the left side, right? Even if it does go to the left side, not enough sodium channels will be present over there to actually maintain this potential, and the threshold towards that side is more, right? So the action potential might be lost, right? Which is why it will move towards this side, right? Once it has started moving towards this right side, it will continue to move towards that side, right? Why will that happen? We will see that also. So first, this region will be depolarized, right? Now, action potential is generated. Now it has to propagate to one side. It is choosing to propagate towards the axonal side, right? Why? Because the sodium ion channel density is still more on the axonal side than the cell body side, right? So that is how it picked first, right? Now, this particular region, the site B will depolarize and the site A will repolarize, right? Now, when the site B has depolarized, it will again move to the next region. Although this will have lesser voltage gated sodium channels as compared to that is present in the axon hillock, right? We talked about this, that site A over here, which is the axon hillock site, has the highest density of voltage gated sodium channels, right? But despite that, from site B, it will move to site C and not site A. Why? Because when it is moving from site B to site C, this site A will be in its refractory state, right? That means that the sodium channels will be inactivated at that time, right? And they will not be able to be activated until this refractory period has passed. At least the absolute refractory period has passed, right? So that is why this action potential cannot revert back and it has to move forward. That is what basically determines the directional flow of this action potential and also makes it unidirectional, right? The similar phenomena will keep happening as it keeps passing on from side B to C, C to D, D to E, and so on, right? So that is how this propagation will continue, right? So there will be two reasons why the action potential will not turn back. One will be the slow closing of the potassium conductances, right? So we have also seen that the channels, once they take a while to get open, but once they are open, they remain open for a longer time, right? So they will close slowly, right? If they will close slowly, obviously there will be that disturbance caused due to potassium ion conductance that will be present. Also, the sodium ion channels will be inactivated, right, during the refractory period. So that is why they will also not be able to help in this depolarization again, right? Okay. Now that we have seen where the action potential is being originated, how it is being propagated, we also need to understand how it will effectively be propagated, right? So there is a condition known as saltatory conduction. Now, what does saltatory actually mean? Saltatory literally means jumping. Right, so it is a conduction where signal is jumping from one end to the other, right? Now, why do we say that saltatory conduction is taking place in our neurons? We will see that. Okay. So, what is happening over here is, yeah. So, when we talk about myelinated axons specifically, right, what will happen is that there will be an insulation of this myelin sheath that will be present on top of this particular axon, right? And this insulation at certain points, there will be a holes in this insulation or it will not be present at those points, right? These points will be known as nodes of Ranvier, right? So what will happen is that once an action potential is generated, right? Because this region is insulated. For example, it is generated over here. This particular region is insulated. It will pass through this region without, any decay or virtually very less decay. So we can consider it to be virtually non-existent, right? Then it comes to this region, which is the node of Ranvier, right? Over here, again, there is no insulation. There will be sodium ion channels that are present over here. Again, over here, it will be negative. Outside, it will be positive, right? Comparatively. And so more sodium ions will flow inside and it will basically, reboot this action potential that is going. And again, it will flow undecayed till the next node of Ranvier, right? So that is what will keep happening, right? Over here, there was an action potential, then there was almost no change throughout this phase, right? After that, there was another, rebooting of that action potential, no change, another rebooting, no change, and so on, right? So that is like it will appear as if the signal is jumping from here, then to here, then to here, then here, then here, right? So that is why it is known as saltatory conduction over here. Then the variations in the properties of voltage-gated channels can increase the signaling capabilities of the neurons, right? So why? Because there is a rich variety of voltage-gated ion channels. Like we were talking about the sodium and the potassium ion channels. Then the gating of voltage sensitive ion channels can be influenced by various cytoplasmic factors. The excitability properties vary between regions of neurons and the excitability properties vary among neurons, right? So thus, these ion channels highly, instruct or they tell how basically there is the signaling within a neuron, right? Now let's talk about the relevance of the action potential. So we have seen previously what is action potential, how it is being generated, right? How what are the ions involved in its generation, how it is basically a summation of the equilibrium potential of multiple kinds of ions, right? And now we have seen how it is being propagated. Now we need to understand the relevance of these action potentials, right? So what is the relevance? When the action potentials will reach the synapse, right? For example, over here, it will result in the release of neurotransmitter which carries information to the follower neurons, right? So action potential, for example, when we are talking about touch, if I'm touching something, it is stimulating some of the neurons, right? Now these neurons are carrying this information into my brain, right? And how are they doing it is that once I have touched something, over there, whatever neuron was there has been a depolarization in that neuron, and an action potential has been generated. Now, this action potential is being carried throughout that particular neuron, and it has to be transmitted to the next neuron so that this signal of that touch can actually reach my brain, right? So that happens via synapses, right? So over here, we are talking about a chemical synapse wherein neurotransmitters are being released by this first neuron into the synapse so that it can stimulate the postsynaptic neuron, right? Yeah. Generally, the graded potentials will not result in the release of neurotransmitters, but there are exceptions, right? So, why are we saying there are exceptions? When we do study the kinds of synapses that are present, we will understand that, right? But in this kind of synapse, where neurotransmitter is being released, right? Over here, grading potential will generally not result in action potential conduction into the next neuron, right? And now we come to actually understand what is term synapses. Before that, let's discuss the scientist Charles Scott Sherrington, who lived between 1857 to 1952, and he won a Nobel Prize in 1932. It was a joint Nobel and his contribution was to basically understand some neural mechanisms, and he also coined the term synapse, right? Over here, I have also kept this logo of this, organization, neurotree.org, which is basically it is the descendants of Sherrington, right? So Sherrington was the person who actually started studying neuro science or neurobiology in the way that we know of it today, right? And he had a lot of disciples, right? And these disciples had further had a lot of other students that they trained, right? And eventually it became, this family tree of sort, and it is known as neurotree.org. And all the people in this can be traced back to Charles Scott Sherrington. Right, so somehow they have gained, some insight through their ancestry from Sherrington. Yeah. So, yeah, after this, what we'll discuss is the work of one of the disciples of Sherrington, that is Otto Louis, right? So, before that, let us just go through this briefly. So, we know that there are multiple kinds of nerves that are coming out of our brain also, right? There are nerves that are coming out of our spinal cord, and there are nerves that are coming out of our brain, right? One of the major nerves that is coming out of our brain is the vagus nerve, right? There are 12 cranial nerves. The 10th number cranial nerve is known as the vagus nerve. It is one of the major ones because it is affecting multiple organs in our body, right? It is a very long branching nerve, and it is affecting multiple body parts, right? So what Otto Louis did was that he basically studied this particular nerve in the frog, right? What he did was that he showed a chemical, right, which was at that time known as the chemicals or the vagus substance, and it was later found out to be this neurotransmitter, that is acetylcholine, conveys signal from vagus nerve to the heart, right? So how he actually found this out was that in his experiment, Otto Louis showed that there is chemical transmission is basically driving the signals from the brain to the vagus nerve or through the vagus nerve to the heart, right? So what he saw over here was that in the first case where we are seeing a donor heart, there was an intact system of the heart, the vagus nerve, that was present, right? And what he did over here was that he removed the fluid sample that was present around this particular heart, right? And after removing this sample, what he did was he took another heart from which the vagus nerve had already been removed, right? And he put this particular fluid sample on that heart. And he saw that this heart, in which vagus nerve was not present, and earlier it was not, responding similar to this heart when vagus nerve was actually present, where it was slowing the heart rate. Once this chemical was actually given in this particular recipient heart as well, there was a slowing of the heart rate, right? So this meant that vagus nerve stimulated the heart in a certain way. Over here, it could be inhibitory in terms of heart rate, right? Because it is slowing down the heart rate. But even when vagus nerve was removed, but only this fluid that was, being supplied somehow by the vagus nerve to the heart was actually introduced. It still could achieve the slowing of the heart, right? So this meant that this fluid might have some kind of a chemical that is actually responsible for the generation or transfer of the signal from the nerve to the heart, right? So Otto Louis basically showed through this that the stimulation or the how the brain speaks to other body parts is basically through chemical messengers, right? Again in 1936, Otto Louis, along with Sir Henry Halliday, received the Nobel Prize for their discoveries relating to chemical transmission of the nerve impulses. Right? Okay, just a moment. Yeah. Um, yes. So once Otto Louis actually published this experiment, although there were other scientists also at that point, such as Henry Dale, who were also basically pitching this idea of chemical transmission, right? Uh, this experiment sparked a lot of debate in 1930s over how the chemical signals could generate electrical activities at the other synapses, right? Which means that in presynaptic, okay, we are understanding that one, it is sending out a signal that is in the form of chemical messenger, and that is going and stimulating the, organ. But if we are talking about basically two different neurons, right? So how could it be possible that there is an electrical signal that is coming that we have already studied is the action potential, right? And then it is converting into a chemical signal, and then it is being transmitted to the other neuron. So how is the other neuron carrying this chemical signal forward, right? And how is it so fast if it is a chemical signal, right? That is what the question was at that time. So there were two schools of thoughts. One was physiological and the other was pharmacological, right? So we'll look at both of these, right? So in the 1950s and 60s, there were improvements in technology that showed that both types of synapses existed, right? So both the pharmacological and physiological kinds of pros and cons of what chemical synapses could do better than electrical synapses, and why electrical synapses should rather be prevalent than chemical synapses. All of that was actually proven when it was actually when different kinds of experiments were done in later 50s, right? 1950s. So what happened at this point was that it was shown that both electrical and chemical synapses were possible. In case of chemical synapses, what was the case was that as we had been studying, whenever there was an action potential, there was release of synaptic vesicles at the end of the presynaptic neuron or the neuron where the action potential had actually generated, and this would release neurotransmitters into the gap between the presynaptic and postsynaptic neuron. These neurotransmitters will go and bind on the receptor of the postsynaptic neuron, and from this receptor, basically, this downstream signaling will be triggered, whereby the action potential will actually be continued in this postsynaptic neuron, right? But in the electrical synapses, what was said was that from the presynaptic and postsynaptic neuron, between this, there were gap junctions that actually connected the cytoplasm of these two neurons, right? From this gap junction, the action potential or the electrical signal itself could directly pass from one neuron to the other neuron, right? So these were the properties of both these electrical and chemical synapses that were described after that. So the distance between pre and postsynaptic cell membranes was around 3.5 nanometer in case of electrical synapse, right? This 3.5 could be bridged by the gap junctions that were present between them. And in case of the chemical synapses, also it was very less in the order of nanometers itself, but it was still higher than the electrical synapse. So it was around 20 to 40 nanometers, right? Then there was cytoplasmic continuity between the electrical synapses, but no cytoplasmic continuity between chemical synaptic cells, right? Then when we saw at the view, or we viewed the ultrastructural components, we saw that there were gap junctions that were actually present, gap junction channels that were present in the electrical synapses, but in case of the chemical synapses, the components varied. They were basically the presynaptic vesicles, the active zones, the postsynaptic receptors, right? Then, in case of electrical synapse, the agent of transmission was an ion current, but it was a chemical transmitter or a neurotransmitter in case of a chemical synapse. There was virtually no synaptic delay in electrical synapses, but there was a small delay in the order of milliseconds, usually 1 to 5 milliseconds in case of chemical synapses. Then the electrical synapses could be bidirectional, but the chemical synapses, from what we have studied just now, were always unidirectional, right? Because there was polarization, depolarization that was going on in this case. Yeah. So let us first go through what electrical synapses were and how they were carrying forward the signal. So it was first discovered in the giant motor synapse of the crayfish. Right now, this crayfish again was another sea-dwelling creature, right? And the experimental setup looked kind of like this, right? In which the presynaptic neuron was basically this lateral giant fiber, this one, right? And the postsynaptic neuron was this one, that is a giant motor fiber, right? So over here we can see both of these, and we can see that there were electrodes that were injected both in the presynaptic and postsynaptic sites, right? And the response was basically recorded. So what was seen was that when an electrical synaptic transmission was done, right, when there was an action potential that was generated, there was a current pulse when there was a current that was given to the presynaptic fiber, or it was stimulated, an action potential was generated, right? And in the postsynaptic potential, you can see almost at the same time such a peak can be seen, right? So it means that there was virtually no delay between the when the peaks of action potential was actually seen between the pre and postsynaptic neuron, right? Which means that the electrical transmission is nearly instantaneous, right? So it is happening very, very quickly, right? There is not even delay in the order of milliseconds that is happening over here. Yeah. Then there was another property in these electrical synapses, right? That even sub-threshold amounts of current in the presynaptic cells can actually depolarize postsynaptic cells through an electrical synapse, right? Why do we say this is because we know that if there were sub-threshold values in case of a chemical synapse, an action potential will not be generated at all. If an action potential is not generated, the synaptic vesicles will not fuse at the presynaptic cleft. And, at the synaptic cleft, there will be no neurotransmitters that will be released, right? But over here, since the two cells are actually connected, right? So even if there are sub-threshold amount of current that are given in the presynaptic cell, it can actually depolarize the postsynaptic cell through electrical synapse, right? It will happen like this. So this is suppose the current pulse that was given to the presynaptic cell, it could not generate an action potential, but it did depolarize up to a certain level in the presynaptic cell. So that means that it can actually also depolarize some up to some amount in the postsynaptic cell, right? Because these two are connected, right? It is not that only after reaching a certain threshold, neurotransmitter will be released, and only then can this be activated, right? It is not like that. Signals flowing directly from the presynaptic to postsynaptic cell, right? So this is what is happening. So the electrical synapses will transport both depolarizing and hyperpolarizing current, but have some rectification, right? So what does this mean? In case of action potential, when we were talking about it, that only happens when the cell is depolarized, right? So if there will be any kind of hyperpolarization, there will be no neurotransmitters that will be released, right? So that is not transferred to the next neuron. But in this case, even if it is depolarizing and that is going towards positive, or hyperpolarizing or going towards negative, right? Both these things can actually be transported to the next cell, right? Because they are continuous with one another. So it will be transported from its higher, gradient location to its lower gradient location. Yeah. Now, when we look at the structure of electrical synapses or gap junctions, it kind of looks like this, right? When we look at it from top through an electron microscope. So this is what it looks like. And you can see that there are these multiple pores that are visible, right? Now, what are these pores, right? So when we look at this, you can see this first membrane is the membrane of the presynaptic neuron, and this next membrane is the membrane of the postsynaptic neuron, right? Normally, there is an extracellular space of around 20 nanometer that is present between these two neurons, but at some junctions, especially where there is an enrichment of these gap junctions or these pores that are present, right? They are only 4 nanometer apart, right? So they are very close by. Now, what happens when they are very close by is that these pores that we can see, right, they align in such a way that they make a continuous, kind of tunnel from this one neuron to the other neuron, right? So there is one set of pore that is present in the first neuron's membrane, second set of pore that is present in second neuron's membrane, right? So it is like we have multiple pipes that are cut. Right, so only when they align properly will there be a tunnel that is created. If even if there were pores, but they were aligned in a way that the pores did not overlap. Right, over here there was a pore, but it went to a place where there was no pore. Right, so it will not be able to cross. So they have to align properly such that these pores are continuous and a tunnel sort of, or this channel is formed by these pores that are put together, right? Uh, and these channels usually had a diameter of around 1.5 nanometer, right? So we know that ions and all can also pass through it very easily. So let's look at the composition of these pores, right? So we said that there are two membranes, there are two sets of these pores, and they are coming together and aligning in such a way that one tunnel is formed or one channel is formed, right? So when we talk about one membrane, right? One membrane has this set of proteins which are known as connexins, and these connexins like this set together, six of them form a pore that is known as a connexon, right? A channel, uh, uh, and it is known as a connexon, right? Now, connexon of the first membrane and connexon of the second membrane come together and interact with the presence of these two extracellular loops that are present in each connexon, right? It helps with interaction with the connexon below it to actually stabilize them in a orientation such that this channel is formed, right? So what will happen is that there are six connexins in a connexon. Each connexin has two extracellular loops that are present for interaction with the other connexon that is present. So each connexon has 12 such loops which have to interact together to actually bind in this kind of orientation, right? And that is why like this is a very very specific interaction that is happening, right? So this is the structure of the electrical synapse. Now, the electrical transmission allows rapid and synchronous firing of interconnected cells, right? How is it helpful? Because immediately the cells will start firing, right? The response that will be generated will be very, very quick, right? So, for example, if we take the instance of this other sea organism that is known as the aplysia, right? In aplysia, what happens is if on this tail region we were to stimulate this tail, right? There was a sensory neuron that might have been present, right? And this sensory neuron would go and it will stimulate multiple motor neurons, right? And these multiple motor neurons, even if it stimulates multiple motor neurons or it stimulates one motor neuron such that it is connected to all other motor neurons also, and it is automatically getting stimulated by this connection, right? And further it is going and stimulating this response, and ink suddenly is being released, right? So that ink being released will confuse the predator, and it will be a protective mechanism for this particular organism to run away, right? So this is what the response will look like, right? So for example, you can see that the sensory neurons might go and stimulate all these motor neurons, right? But these motor neurons are also connected. Even if the sensory neurons were stimulating all motor neurons, it is not uh, always possible that the reaction will occur at the exact same time. But if these motor neurons are also connecting it in turn, right, via this electrical synapses, then this response can occur at exactly the same time also, like we saw in that action potential peak, that there is no delay between the response, right? So that is what is happening over here. From all these neurons, A, B, and C, the action potential is reaching a peak at the same time. It is following the same curve, and thus the summated, response is able to release the ink from the ink gland, right? Yeah. So that basically brings us to the end of the electrical synapses, right? Now let us come at chemical synapses. So why will we also discuss about chemical synapses? Because these are the more abundant kinds of synapses that are present. Now, even though we said that electrical synapses are faster, right, and they are also efficient in transporting information, why are not more electrical synapses present? Because it also has some cons, right? We did say that it is not always important that the electrical synapse reaches an action potential for the potential to be passed on to other neurons, right? So it can even survive on graded potential. So sometimes it might be possible that the stimulus is not very harsh. Despite that, our body is producing a very harsh kind of response to that very mild stimulus, right? We don't want that because we don't want our energy to go in waste, right? Or in vain. So, there's another reason also. We saw that chemical synapses will have transmission of the signal unidirectionally, right? So, if I am stimulating over here, the signal will go through this path into my brain, right? But in case of electrical synapses, what will happen is that if I'm stimulating over here and this was an electrical synapse, it can go to synapses nearby. Again, when it comes till here, it can go over here, right? Then it can jump over here. It can come back also, right? So it will not be a very efficient kind of response, right? I don't want it to be wandering over here, the response. I want that action potential to be taken straight to my brain so that my brain can tell me what to do, right? For these reasons, basically, chemical synapses exist, right? So we have talked about chemical synapses multiple times now. This is the easiest representation of the synapse, wherein there is a pre-synaptic neuron which has synaptic vesicles, which basically on the initiation or on the in the at the presence of an action potential, these synaptic vesicles can actually go and it can fuse with the terminal sites of this presynaptic neuron. And after fusion, the whatever is contained in the synaptic vesicles, which are neurotransmitters, will be released in the space that is present between the two neurons. Right, this space is known as synaptic cleft. Neurotransmitters will be released into this space, and then they will be attached to the receptors that are present on the postsynaptic neurons, right? And these receptors on interaction will generate, the appropriate signal so that this action potential can be transmitted forward, right? So that is what chemical synapse is, right? Now let us speak about these main regions. So we talked about synaptic cleft. It is basically the region separating the presynaptic and postsynaptic cells at a chemical synapse, right? This is synaptic cleft. Then the neurotransmitter that is shown in red over here is a substance that is released from the presynaptic terminus that will bind to the specific receptors on the postsynaptic cell membrane, right? Then there will be synaptic vesicles that are seen over here. The presynaptic terminal contains discrete collection of these vesicles filled with thousands of molecules of a specific of a specific transmitter. Then there will be some active zones or regions of the membrane specialized for release of neurotransmitters, right? So this will be the active zone. If we look at the electron microscope image of this synapse, this is what it looks like. Right over here, we can see some distance between the two neurons, but in an electron microscopic image, we can actually make out how big this difference is, right? It is very, very tiny. Like we talked about, it is in the order of nanometers, right? It is around 20 to 40 nanometers, and this image is in the scale is in zero point 0.5 microns, right? So it is, almost tens or hundreds of nanometers, right? So that is the reason why we can't see them as very far apart, but we can actually make out that this is the axon terminal, these are the synaptic vesicles, this is the synaptic cleft, the very tiny narrow space that is present between, and these synaptic vesicles, the ones that are coming on the axon terminal and fusing with this cleft, will release neurotransmitter that will be present over here. This is the postsynaptic neurons dendrite, right? And it will be able to have receptors on its membranes which can, bind to which the neurotransmitters can come and bind such that signal will be, propagated along this particular dendrite into this particular neuron as well. So these dark arrows will indicate the active zone where basically the neurotransmitter is being released. Okay. So let us look at how processing of the signal takes place at the chemical synapse, right? So firstly, again is the presynaptic cleft or presynaptic neuron, the synaptic cleft. There are synaptic vesicles over here, right? There are some channels over here. There are some channels at the end of this the other neuron that is also present, right? So there will be action potential that will cause voltage gated calcium ion channels to open and allow influx of calcium, right? Because whenever there is action potential, there is depolarization. The calcium ions are positively charged, right? And they are present more abundantly extracellularly, right? So both chemical and electrical gradient will push them inside, right? They will want the calcium ion to flow inside. So whenever there is an influx of the calcium ion channels, by calcium ions by the opening of their channels, right? What will happen is that these calcium ions will go inside and the neurotransmitters in response to that will be released outside, right? Because the abundance of calcium causes synaptic vesicles to fuse within the membrane, with the membrane and release the neurotransmitter. Once this neurotransmitter is actually released, what will happen is that the presynaptic nerve terminal over here, will basically, sorry, the neurotransmitter will that is released through this presynaptic nerve terminal will diffuse to the postsynaptic membrane where it binds to specific receptors and the results in opening of these channels, thus changing its membrane potential. Okay, so this is a response that we are going to get. Now, once this response has been achieved, we need to know that it is not going to happen that the neurotransmitter is continuously there in that synaptic cleft, right? Because if it is continuously going to remain there, it is continuously going to go and it is going to keep on binding to these receptors that are present on the postsynaptic vesicles, right? Postsynaptic terminal, right? And if that interaction between these neurotransmitters and those receptors, if that keeps occurring, then this neuron will always remain activated, right? There will be no difference between its active and inactive state, right? And we don't want it to be continuously active. That is a very energy intensive process also. And also, when the next signal comes and we need to transport that next signal, it will not be able to do that because it is already engaged, right? So what needs to be done is that this neurotransmitter that is left over at the synaptic cleft actually needs to be removed, right? So there are various mechanisms for its removal. One of the major ones is the reuptake mechanism, right? So in reuptake, what happens is that usually there are not just receptors that are present over here on the postsynaptic, neuron or the dendrite of that neuron, but even on the axon of the original
Transporter proteins on
presynaptic.
the precentriminal that we said.
Yeah, yeah. Can
I'm
you
so
can you
sorry.
say that again?
Oh, sorry, sorry. Kindly ignore, ma'am. Let's go ahead.
Okay, okay. Yeah. So as I was saying, what also happens is that on the end of the first neuron, right, the synaptic neuron, uh, the axon of which was actually the responsible for that synaptic vesicle fusion and release of neurotransmitters. That end also contains certain channels that can actually help in replenishing whatever neurotransmitters have gone out back into this first neuron's axon, such that it will be reuptaken over here by these protein uptake channels, and again it will be pumped into this axon terminal, and they will be surrounded by a synaptic vesicle again, and they will remain again in this one. So what this does is that it helps in recycling of the same neurotransmitter again, right? So it does not have to be broken, its chemical composition does not have to be changed. What it does is that after some time, when this depolarization over here is over, right, this channel will open up. And because there is more neurotransmitter outside now, and this channel can allow some of it to come back, that is what it will do, right? So this reuptake of this neurotransmitter will happen. Now, another interesting point is that whenever, for example, there are multiple conditions in which neurotransmitters are lesser in certain people. So there is a chemical imbalance of neurotransmitters in some people, right? This chemical transmission might be of various kinds of neurotransmitters. It might be serotonin or dopamine, or maybe acetylcholine, or etc., right? So in this case, one kind of therapy that is given to such people is the use of SSR. So that is so that the reuptake of these neurotransmitters from the synaptic cleft will be amplified, so that whatever little neurotransmitter we have, we don't lose that, we can use it multiple times, right? So that is one kind of therapy that is given to people that have less of this neurotransmitter that is present. It might be due to neurons that actually make this neurotransmitter are basically going down in number, right? For example, in case of Parkinson's, the dopaminergic neurons are dying. So there will be less and less of dopamine that is present, right? So we need whatever is present to be utilized to its fullest, right? So that is one kind of therapy also. So, coming on to the second kind, which is enzymatic degradation, right? In this, acetylcholine, for example, is usually, removed from the synaptic cleft in this manner. What happens is that there is an enzyme that is specific for that neurotransmitter. In case of acetylcholine, it is cholinesterase that is doing it, that is present in this synaptic cleft, right? And after a while, after a while, when this neurotransmitter is present over there, it will not just go and interact to the receptor, the free ones will be acted upon by this enzyme, and it will be degraded over there, right? And then the degraded material will also be removed. Then the third one is diffusion. So in diffusion, what is happening? There is moving out from the cleft. These neurotransmitters are moving out of the cleft, and they are eventually getting absorbed or excreted out, right? So that is like, because earlier they were enriched over here. Now there are other regions where they are less in number, so they will move to those regions, and eventually either they will be degraded, or they will be excreted out, or they will be absorbed by some other neuron that is present over there, right? After that, we come to this postsynaptic potential. So till now, what we have read is what is a synapse, right? How basically this synapse is, transferring its energy or its potential, whatever information it has, how is it transferring, right? So whether it is chemical or electrical, that is dependent on the presynaptic neuron mostly, like, right? Then we also saw that whatever neurotransmitter is being, present in that synaptic cleft, thereafter, after the action potential has already propagated, what is happening with that, right? That also we have studied. Now we need to see that once this neurotransmitter has bound to that receptor, how postsynaptic potential is actually working, right? In this case also, and in the electrical electrical synapses also. So just a moment, let me just come back. Just a moment. Yeah. Yeah. So what is happening at the postsynaptic potential basically? It is how basically the activity that is being caused in the postsynaptic neuron is being achieved, right? So what happens over here, whatever signal we are receiving at the postsynaptic end, that is the dendrite of the postsynaptic neuron, that is being converted into a potential energy that is known as postsynaptic potential, right? So this postsynaptic potential could either be excitatory or inhibitory in nature, right? That will depend on whether we are calling it as EPSP or IPSP, right? So whether it is excitatory or inhibitory, it will always depend on which ion channels are being opened by the neurotransmitter, right? So, for example, the neurotransmitter is, GABA, right? So it will open the kind of ion channels that will hyperpolarize the membrane. So it will be inhibitory in nature. But if it was glutamine, for example, or it was acetylcholine, it will most probably open ion channels which will depolarize the membrane, right? If it will depolarize the membrane, then it will be excitatory in nature, right? So, if we talk about an excitatory postsynaptic potential, right. If, for example, we are getting a response over here, after a slight delay, the response will be achieved. And if this response reaches the threshold, an action potential will be achieved. But if it fails to reach that threshold, action potential will not be released, achieved, right? The synaptic delay can be used to tell whether two neurons are connected to each other, right? So, for example, we are saying that there was a presynaptic action potential over here, and this much delay was there. After that, postsynaptic potential was seen, right? Now, there were maybe three such excitatory potentials, postsynaptic potentials that I measured from three different neurons that were in that vicinity, right? So, depending on what is the amount of delay in each one of them, I can, identify whether this presynaptic action potential is directly linking to that postsynaptic neuron or is going through it via some other neuron, right? So, for example, one had this much of a delay, one had this much of delay, and the third had this much of delay, right? So, I know that this presynaptic is going via this first to second to third, right? That is why the third one has that big of a delay, right? Okay. So, again, we did ponder upon this question a little bit about why to use chemical synapses when electrical ones are also present and they are directly, they don't even need neurotransmitters. They can directly transfer action potential, right? Even an action potential is not really required or necessary to, build or transfer information, right? But still, we do use chemical synapses. Now, why? So apart from the reasons that we had already said or discussed, there are these other reasons, right? So one is that the amplification is tunable, right? Which means that how many synaptic vesicles are coming and binding to this particular site or this particular surface, how much neurotransmitter is being released, how many receptors are being localized to the dendrite of this postsynaptic neuron, right? All this is modifiable, right? And from response to response, it will vary. So it will probably happen that the same machinery is being used even when I'm using my phone and I'm using my laptop versus if I get burned, right? Because all of this is touch response, right? So when I'm using my phone, I'm not in danger of getting burned or anything, right? So the same mechanism is being followed, but maybe the amount of receptors that are coming over here, attaching over here might be lesser, right? The amount of neurotransmitter that is being released might be lesser. While if on that very finger, if I get burned, right, all of a sudden, it will stimulate a different kind of response. It will be much stronger. I am expecting more neurotransmitter to be released, more of these receptors to be localized at this postsynaptic, terminal, all these things, right? So this amplification is tunable. And because this amplification is tunable, how weak or how strong a synapse or a synaptic response will be, will actually, uh, dictate how strong of a memory, for example, can be formed, or how light of a memory will be formed, right? That is one. The second is flexibility. Flexibility in the sense that once a neurotransmitter is released, it is not that only this particular neuron is present in its vicinity. There will be maybe five other neurons that are present in its vicinity over here, over here, over here, over here, right? And all of them can be stimulated by that particular neurotransmitter, or it might be possible that the one that is directly facing it might not at all have receptors for this particular neurotransmitters, and only the side ones are being stimulated. So it is very flexible in that regard, right? And there is obviously filtering, because only if you have receptors for such for that particular neurotransmitter will you actually receive the signal. Also, only if action potential is actually being generated will that neuron be able to transmit its information to the next neuron, right? So all these factors are there, due to which chemical synapses are actually still used, and it is widely present in our system. It is the most common kind of synapses that we have, right? Then let us read about some common neurotransmitters, right? So one of the most common neurotransmitter, or the most common excitatory neurotransmitter actually is glutamate, right? This is the structure of the glutamate. Then GABA, or gamma aminobutyric acid, is the most common inhibitory neurotransmitter. Then acetylcholine is also a very common neurotransmitter. It is usually present in the motor neurons, right? The neurons that are mostly also attaching to the muscle. Then there are also some other common neurotransmitters such as dopamine, serotonin, and noradrenaline. These are involved in large scale modulation of the brain. And thus they are also known as neuromodulators, right? Then the neurotransmitters can open channels on postsynaptic neuron directly or indirectly, right? Why do we say that? Because in case of inotropic receptor, for example, what will happen is that the ligand is going at, which is a neurotransmitter, is going and binding on a particular receptor, and this receptor is, for example, capable for a particular kind of ion to pass through it. And it is because this binding is aiding this receptor to now be open like this, right? And now this particular ion for which it can allow the flow can actually pass through. So it is happening instantaneously, right? The neurotransmitter is going and binding this closed channel. This closed channel is opening up, and now this particular ion can actually pass through, right? For example, sodium ion, right? So it will directly affect the postsynaptic potential, right? The other one is indirectly affecting the postsynaptic potential. So in this case, for example, there is a metabotropic receptor, such as the most common ones that are present in our body, is GPCRs, or G protein coupled receptor. In this, what will happen is there will be a receptor to which the neurotransmitter will go and bind. This receptor on its intracellular side or the cytoplasmic side will have a G protein that is coupled with alpha, beta, and gamma subunits. Right now, its subunits will dissipate when this neurotransmitter comes and binds. Right, either the alpha one will act or the beta gamma one will act, and it will basically go and stimulate an ion channel that is present nearby to basically open so that ions can flow in, right? So in this, it is not directly the ion channel is that is being affected. Another receptor is going and being bind upon, right? After that, that receptor is going and it is signaling other chemical messengers to go and open that other channel, ion channel that is present far away, okay, or maybe even nearby, but not exactly on that particular receptor, right? So then that channel is being opened, and from there the ions can actually flow in. So it is indirect and it is a little slower, right? Because it is indirect, right? Then let us see the receptors for common neurotransmitters, right? So, for example, there are glutamate receptors, which are generally for most of these glutamatergic neurotransmitters, right? There are inotropics for NMDA receptors, which is N-methyl D-aspartate, which the neurotransmitter is actually responsible for learning, memory, and synaptic plasticity in the brain. The AMPA receptors, which is basically responsible for fast excitatory synaptic transmission in the CNS, and kainate receptors, which are basically involved in short-term and long-term plasticity and long-term potentiation. And these kainate receptors are also the ones that go haywire in epileptic brains, right? Also, all these cation channels are permeable to potassium and sodium, and equilibrium potential for these particular channels is near 0 millivolt, right? Then there are also metabotropic receptors, metabotropic glutamate receptors, which are GPCRs. So all these are majorly excitatory receptors, right? Then come the GABAergic receptors, which are can also be both inotropic, that is GABA A receptor, and metabotropic, that is GABA B receptor, right? So the GABA A receptor is permeable to chlorine, and the GABA B receptor is permeable to K plus, right, or potassium. And this receptor, depending on where it is present, could either have an inhibitory potential or an excitatory potential, right? Depending on what is the resting membrane potential at that particular site. Then we talk about synaptic integration, wherein a neuron will receive input from multiple other neurons, and its activity will depend on the net input that is received, right? Why are we saying this? Because there will be some neurons that will be given excitatory stimulus, and some others that will be giving an inhibitory stimulus, right? So the ones that are actually giving an inhibitory stimulus are the ones that are actually going to, uh, determine whether action potential over there will be achieved or it will not be achieved, because we are going to negate all the excitatory stimulus that is being released, right? Then the presynaptic neurons may give excitatory or inhibitory inputs to postsynaptic neuron. A single postsynaptic neuron may also be able to receive both excitatory and inhibitory inputs, right? For example, if we talk about the knee-jerk reflex again, we saw that there are both kinds of these inputs that are present. One is the excitatory input, and the other is the inhibitory input, right? The excitatory input will be towards the quadricep muscles, and it will basically stimulate this quadricep muscle to contract so that the knee is, jerk forward, and the biceps over here will basically have an inhibitory response, so they will not contract such that it is not pulled backward, right? So what will happen if we look at the experimental setup for recording this stimulus? What we will see is that there are sensory fibers, right? These sensory fibers that are coming from these muscles, right? Once this jerk, this stimulus is given, this sensory fiber will take it via this part to the spinal cord, right? This basic ganglia region in the spinal cord. And over here, what will happen is, you can see there are two circuits that are going, right? Is this one direct circuit, which will basically conduct the sensory information. It will go to this, motor neuron, and this motor neuron, because of the multiple sensory information that it is, because of the sensory information it is receiving from multiple excitatory neurons, it will basically generate an action potential, right? A little while later, after receiving the sensory information and after generating this action potential, it will send it back so that the muscle is contracted, right? So this is a excitatory potential. Now there is this other mechanism that is going, that is indirectly, where the sensory information is coming, it is being passed on to the inhibitory neuron, and this inhibitory neuron is basically forwarding this to the motor neuron, right? So what will happen over here? Over here, this inhibitory neuron will basically go and it will, give a inhibitory postsynaptic potential to this motor neuron, right? Once it is an inhibitory postsynaptic potential, what will happen is that after this interneuron actually had a action potential, it was able to transmit it to the motor neuron, and after a little while, after a short delay, it will go into hyperpolarization, right? And it will have a negative potential or inhibitory potential, right? What will this do? That it will delay the uh, uh, delay the contraction of the lower muscle. That was the bicep over here, right? This bicep or the flexor muscle, it will delay its contraction, right? Even if another jerk is, given to this muscle, all of a sudden it cannot, achieve an action potential, because now the action potential is farther up, because instead of being minus 70, now it is probably minus 90. So it has to come over here. It needs more value to come over here, right? So that is why it is inhibiting the next response, basically, right? Then, if you look at the firing of a neuron, it is basically reduced by the inhibitory input, right? So there is no firing that will occur after an inhibitory neuron, but it will reduce the actual firing that is happening, right? So it will cause it interference, right? So this is what it looks like when we do receive an IPSP. So, yeah. What else? So this is the kind of thing we talked about when we were also talking about that action potential. Why action potential is generated in the axon hillock region, not in the cell body, not in the dendrite, right? We talked about this at that point also, because the threshold is very high at this time, right? Even though the potential actually might also be higher over here, right? But the threshold is also very high. So only when the threshold decreases because of the increased abundance of the sodium ion channels that are present over here can we actually work this out, and can action potential actually go higher, even though the peak synaptic potential is actually going lower as it is coming from the dendrite to the axon hillock region, right? So yeah, if we talk about the temporal summation, that is how much time basically it is taking for this, signal to be transmitted. We can see that the two synaptic inputs at different times can produce a summation effect if the delay between the two is relatively small to the membrane time constant of the neuron, right? So, for example, if there was one stimulus that was given, and immediately another stimulus was given, such that the first had not died down completely, right? If it had died down completely, it will come back to normal, and then it will start again from this baseline, right? But if this had not come back to normal, or it had not come back to the baseline altogether, and it had been stimulated again, such that from the middle only you give that much only again, such that it crosses the threshold, right? So uh, in that case, action potential can be achieved, right? So that is known as temporal summation. Now, similar is the spatial summation, right? So, for example, if the length constant had not died down, it had not come below 37% or to the 37% of the original value, and before that, another threshold was given, right? From here to here, it had not died down, but another threshold was given, right? Another sub threshold was given, but this was also at certain level, right? So after this, that much level itself would be enough to actually attain a threshold, and a signal can be achieved, right? Yeah. Then there was a short talk about the kinds of synapses. One was the axodendritic synapses that was present, right? In this, what was the which is the most common one is that there are dendrites, right? And there are some branches that are even coming out from the dendrites. And usually these synapses are being formed between the axon terminals and these dendrites and the spines that are coming out of the dendrites, the spine synapses, and the shafts of these dendrites, so the shaft synapses. But there are two other kinds of synapses also, wherein the axon terminal can directly form a synapse with the other axon terminal of the other neuron, which is axoaxonic synapse, or it can form a synapse with the cell body of the other neuron, that is known as axosomatic synapse, right? So this is what it looks like. So the synaptic contact can occur on the cell body, the dendrites or the axon of the postsynaptic cell, right? So this either of this can actually happen. So that brings us to this final part, that is the sensory systems. So let me just check the time. We have 15 minutes. Let's see how much we can cover in 15 minutes, right? Because it will be hard to finish this, I think, in 15 minutes. So whatever part I'm not able to discuss, I'll pick that up in the next session, maybe. If we have a discussion session, or I'll just post whatever, summarized view of this documents I have created. I'll just post it on that documents in NPTEL, right? Yeah. So when we talk about the sensory systems, there was this theory earlier that our brain is basically like a blank slate, right? So there were some empiricists such as John Locke, David Hume. These are very famous philosophers, authors, poets, right? That you might have heard about in your history books, in your literature books, right? So these people were empiricists who actually believed that all knowledge is obtained through sensory experience. At the time of birth, the human mind is a blank slate on which experience leaves its mark, right? But on the contrary, there is strong evidence that brains are pre-configured to respond to certain types of stimuli in certain manners, right? For example, liking for sweet taste. For example, all of us do because of how our ancestors have actually grown and survived. All of us are naturally inclined towards sweet tasting things and towards carbs and fats, right? So that is our natural inclination. Not all of us, by birth will be like vegans or keto diet following people. Not all of us will be like that, right? Because that is how our brains have been configured, right? Through and it has been passed on epigenetically to us, right? So, yeah. So first, let us begin with the discussion of the visual stimulus, right? Which is one of the most dynamic kind of sensory stimulus that we receive. And there is a very large region of our brain that is dedicated for visual perception of both, right? So when we receive the electromagnetic waves of different frequencies, we perceive them as colors, right? So these are simply waves of different frequencies. My pointer is emitting electromagnetic waves of certain frequency. That is why you are seeing it as red, right? Otherwise, in time and space, it might not have been that color, right? It is how our brains are perceiving as, right? A red color, my pointer, laser pointer, right? So this is the range that humans can actually visualize. It ranges from 400 nanometer, which starts from around that violet indigo range, to 700 to 800 nanometer, which is a red orange range, right? So this is the wavelength that we can actually see, right? Between this wavelength, right? Yeah, this is basically the visible range, as we call it. Below this, we call it as X-rays and gamma rays. Above this, we call it as ultraviolet, infrared, microwaves, radio, and TV waves, right? Yeah, so that is that. So between this around 400, 300 to 400 nanometer visible range is all the different kinds of colors that we are actually able to visualize, right? So when we even if this is the case, even in terms of sound, right? We do not really hear a particular note or something, right? It is just the vibrations in that particular instrument or my voice is causing in the air around me that is being transmitted to the microphone, that is reaching your guys speakers, and that again is transmitting it in the form of waves into the air around you or into the earpieces that you are wearing, right? So that you can hear my voice in a certain way, right? So it is just due to vibrating of different objects at different frequencies that we can hear sounds or words of music, right? So let's talk about the different kinds of senses. There are five major sensory modalities that we know of, that we have already studied about since we were kids, right? These are vision, hearing, touch, taste, and smell. And out of this, touch is the most widely present one, right? Apart from this, there are some other senses that we are also going to talk about over here, right? So, what are these senses? These are basically itch, temperature, um, pain, proprioception. Proprioception is something we talked about in the PYQ session, right? And the vestibular sense of balance, etc. Right? So these are other things also that we perceive, right? Then the sensory receptor is basically a specialized neural structure on which the external stimulus is received and converted into an electrical energy. Right? So that is what a sensory receptor is. And how is the stimulus transduced? It is by a process which a particular stimulus energy is converted to electrical signal in the receptor. Right? So that in that process is known as stimulus transduction, the conversion of this chemical energy into an electrical energy, right? Or mechanical energy into an electrical energy. So, we also know this as, the higher functioning of these sensory organs as modality. So what is modality? It is represented by the ensemble of neurons connected to specific class of receptors, right? Such ensembles of neurons are referred to as sensory systems. For example, there is a somatosensory system, which will, be responsible for the touch-related senses, the visual system, which will be responsible for the sight-related senses, the auditory-related senses, vestibular, or the hearing related again, or the balance related senses, rather, the olfactory system, the odor related ones, and the gustatory system, or the taste related ones, right? So there are different sets of neurons that are basically responsible for different ones of these senses, right? And it is also because these neurons are actually working as in response to electrical stimulation, right? So even if artificially those particular neurons that are seeing that particular object in front of me as a plant, if that is actually stimulated artificially also in that very manner, then even if there wasn't a plant and those particular receptors were being stimulated in that very manner, and the same kind of electrical firing was going on, I might still see a plant over there, right? So an artificial stimulation of particular sensory neuron basically elicits the same sensation, right? Uh, which is why the phantom limb syndrome people also actually have enormous pain, right? So what happens in a phantom limb is that maybe you do not have a particular arm, but despite that, like it has been amputated due to some disease condition, due to some injury, due to some kind of accident, right? Your arm might have been amputated, but the patient of a phantom limb disease, what that patient feels is that the finger, like particularly the fingers or the wrist, is aching like anything. Now, the fact is that the person does not have that wrist, right, or that finger, but that that particular region is still aching for that person. Why is it so? Because maybe the nerve ends from here are gone, but the higher order nerve connections that they were actually stimulating still have loose nerve ends that have similar electrical signals that it is getting, right? From some kind of imbalance that is going on, some kind of connection loss that has happened. It is still being stimulated in the same way, which is why the mind is thinking that I do have a wrist that is broken, or I do have fingers that are aching like anything, right? Although my arm is amputated, right? So that is also because of this phenomena only. Yeah. So this is a very long chart that I have kept, but I'll just read it once and you can go through it yourself, right? So over here, what we talk about is the sensory system that is involved, the modality that is present, the stimulus that is being given, the receptor class, and the receptor cells, right? So, for example, in the visual sensory system, the modality or what we are actually doing is vision, like perceiving vision, right? The stimulus is the light or the photons that are present. The class of receptors is basically the photoreceptors, and the receptor cells are the rods and the cones, right? Similarly, if we talk about auditory, the modality is hearing, the stimulus is the sound or the pressure waves around us, then there are mechanoreceptors that are present, and the cells that are perceiving is the hair cells in the cochlea, right? In the vestibular ones, there is this head motion that is going on. The stimulus over here is gravity and acceleration and head motion. Again, there are mechanoreceptors, and the receptor cells are the hair cells in the vestibular labyrinth, right? That is also in the inner ear only, right? Or connecting to the eustachian tube. Then there are multiple kinds of somatosensory responses, such as touch, wherein the stimulus is skin deformation and motion, right? For example, even if I'm putting a comb on my skin, the comb teeth will deform my skin in some way, right? Even a minor deformation can be read by the skin receptors, right? And these are also mechanoreceptors. The cells are present in the skin in proprioception. Again, mechanoreceptors are involved, and the receptor cells are in the muscle spindles, the joint capsules. That is why we don't really have to look at them. We know that the capsule is moving and the joint is moving, right? Then the pain also has different classes, receptor classes. It could either be chemoreceptor or thermoreceptor or mechanoreceptor, right? All of the tissues except CNS have this receptor, the pain receptor, right? Then the itch, the stimulus is histamine, right? Because histamine is that chemical that will cause that sensation of itching, right? Then the receptor class is the chemoreceptor, and the receptor cells are on the skin, because you're not going to go and itch in your stomach, maybe, or nothing is going to itch in your stomach or on your heart or on your lung, right? It will just be on the surface of your body. And then there will be visceral, which is not painful. There could be different kinds of receptors, thermomechano or chemoreceptors, and the receptor cells are usually in the GI tract, the urinary bladder, the lungs, etc. Okay. Then there could be gustatory sensory system. The modality is taste, stimulus is chemicals, then chemoreceptor is involved, and they are present on the taste buds, right? And the final one is olfactory, wherein the modality is smell. Odorants are the stimulus, chemoreceptors that are present in the cilia on the olfactory sensory neurons are actually responsible, right? Then we come to the tuning of the sensory receptors, right? Now, this topic is a relatively long one if I started, right? So, what we can do is from this topic, that is tuning of sensory modalities or sensory receptors, we can pick up in the extra session that we are going to have, right? And I'm sure that we have not yet decided upon a time for that session. So I'm not sure whether we are going to have it on next Sunday itself, but we'll try to keep it, right? So as soon as we get that option from NPTEL. So for now, what I can do is I can stop sharing the screen. When I do share this particular presentation with you, I'll do it as a whole. I will not cut out this part. But as far as discussing this part is a concern, that I'll do in the next, session. So yeah, I'll just stop recording. Thank you all for bearing with me this long. I hope like you do not have any doubts pertaining to this. If you do, you are free to ask. I will just stop the recording, right?